there are two industry talk, and we will move on to the first talk by Subhadeep Mukherjee. He is from BD Life Sciences, Biosciences, and he is going to talk on BD technology and innovation facts discovered. Please.
Good afternoon, everyone. Can you hear me well?
Yeah.
Okay. Just allow me some time to connect the system. Again, good afternoon and a warm welcome to this session from BD Biosciences. I would like to thank first Professor Alok and the organizing committee to give us an opportunity to talk here. It's an absolute honor. So this talk is about the BD technology and innovation. So when I was a customer of BD, I was on the other side during my PhD days. I was always looking at BD, that word they are bringing new in the flow cytometry space. Because they are the leader, right? And now I have the privilege to present one of that, which Facts Discover. It's not only an instrument what I'm going to present, but also groundbreaking technology that we'll see now. So let us start with what it is and what innovation it is bringing in. So right now, if we review the two common ways or broad ways of looking at a single cell. So one, we find in microscopy, right? That gives us a spatial or morphological information of a cell. But if we want to characterize deep, want to go deep down into the cell functionality, the identification of that, then you might need flow cytometry. So you can go higher into the dimension in terms of how many receptors that cell is expressing. If that cell is a very rare type, lying deep into or masked into the other cell types which are broadly expressed or broadly represented, then you need flow cytometry to find that. But these two technologies right now are complementary, right? And we do have utilization or benefits of both of them. So, of course, the research is going on in this space that whether we can leverage a technology, we can come up with a technology which leverages the benefit of both the microscopy and the flow cytometry. So we can understand the spatial information as well as the immunophenotyping, what we call in terms of immunology or characterizing the cell. So this paper, which was came into the science cover in early this year in February, that talks about imaging, using the imaging information for cell sorting. So this is something new, this is something innovative that came up. It is a joint publication from EMBL and BD. So imaging flow cytometry is not new to market. There are imaging analyzers which actually utilizes the flow technology and also gives the imaging information. But you cannot take a decision for sorting the cell post-analysis so far unless this publication came in, which talks about the real-time imaging. So you can take a decision for cell sorting. So you can do your downstream experimentation, that can be a sequencing or any culture further. If you making a hybridoma, you are going for any other drug development techniques, whatever you want to do post identification and sorting of the cell, you can take a decision for sort. So this is new. This is the innovative thing that came in of taking a sort decision based on imaging. BD calls it cell view imaging technology. So you can combine this, or enjoy the benefit of imaging along with flow cytometry. So what you are getting here, the high-speed, high-parameter flow cytometry intact. You are getting the high-content imaging data during analysis, and finally, you can use that imaging data along with or separately the way you want it with the flow cytometry data to take a sort decision. How does interface looks like? So this on the left side of this like slide what we are seeing in the light showed blurred. So this is a very common flow cytometry basic slide or kind of a data what you see in the dot plot, right? You are seeing three different populations of lymphocyte, monocytes, and the neutrophils. So here the extra what we are getting is the image wall on the right side of that of this slide. So that image wall appears simultaneously with this dot plot. So when you are analyzing the cell, when the cell is still moving through the flow cell, it is interrogating by the laser. You are getting this individual picture of each and every cell the laser is interacting. So you can see, you can visualize, you can see the spatial information or morphological information of the cell along with this flow data what you regularly get out of a flow cytometer. So what advantages it broadly brings up? Primarily, it is expanding the scope of flow cytometry technology utilization beyond immunology, because flow cytometry is something, a term usually related to immunology. Immunologists are using it the most. But now with this morphological information coming in through imaging, now it can be utilized in the cell biology as well. If you are working in say different translocation assays, intracellular signaling like nuclear translocation assays, you will be able to see it, you will be able to visualize it along with the regular flow data what you do. If you are looking at any subcellular organelles, that you can visualize, you can take a picture of that. And also different internalization assays, cell-cell interaction, immune synapses, I have a data for that I will show you. Those things which were so far we were not able to measure through flow cytometry, or maybe the measurement was indirect, now we are able to see it or visualize it directly. And the last one is of course on the DNA and cell division analysis. DNA content analysis, ploidy, is something researchers are regularly doing with flow cytometry, but so far with a conventional flow technique it is not possible to look into the different stages of cell division, right? But here, as you can visualize it, you can see the different phases of cell division along with the ploid information. So that's what we are adding in with this. How the technology works. So basically this technology is adapted from telecom industry, telecommunication, broadband. We call it orthogonal frequency domain multiplexing. So it is a very same technology, the way a broadband router is connecting to 5 different systems. So laser is also modulated at different frequencies. So it's not a single laser beam which is hitting the cell. There is a modulator associated with the system which is modulating the laser at different frequencies, and when the cell is passing through the laser at different, as you can see, this F0, F1, F2 all are hitting the cell in an overlapping manner, and we are getting this waveforms, what we call it, which is generating the image along with the flow data. And how we are collecting this image data, which is generated simultaneously, we are collecting it in a form of a TIFF, T format, and all the TIFF together, because maybe one cell are getting 1000 like same pictures of waveforms. So all are combined together in a GStack file. So when you are getting the output, you are getting this file .fcs is a regular flow cytometry data raw file what you get out of a flow cytometer. Along with that, you are now getting the GStack which gives you the imaging data. So if you want to go further down, you can utilize the FlowJo software to analyze this imaging data as well. Now let us see that what kind of data we get out of this and what benefit this gives you. So one is on the mouse over imaging for scatter detection, right? So we in flow cytometry, when we are running any sample, we are taking a decision for sort. First we discriminate that these are our singlets and these are our doublets, means whether two cells are fused together or not. So this doublet information, so far we are taking that call based on the forward scatter and side scatter data out of a dot plot. But now as you can see the cell, you can, like the way you are seeing on this right side of the plot, you can see whether you are looking at two cells fused together or you are looking at a single cell. So easily you can take a decision that whether you are looking at a singlet or a doublet. You don't need to do any indirect measurement or decision making criteria based on the dot plot. On the bottom of the plot, what you can see is a measurement, what we call the index sorting. If you like to sort one single cell in 96 well or 384 well plate, it is possible with any regular flow cytometer sorter nowadays. But what it is adding up, that if you are sorting say 96 cells on 96 well, okay, of a plate, then you can see each of this cell which are deposited in the well, and you will be able to understand whether you have sorted the right cell. So you are more confident if you are taking further those cells for culture or sequencing, that you have the right kind of cell in that well deposited. And so that's the addition, that's the quality metrics it is adding on. Tumor interrogation now become more easy, as now you don't have to look into the TILs based on their markers. Here is a mixture of mix of the TILs and the cancer cell, and based on that image wall, you can clearly see that the spatial information, the morphology of cancer cells are very much different. That is, of course, that is obvious from the lymphocytes, which are more circular in nature. So you can, without any labeling, just with a calcium dye, which is a live dead dye, analysis dye, so you can take a decision that whether you want to sort TILs or the cancer cells. Cell-cell interaction studies, mostly the immunological synapses. This is really a benefit which we are adding on with this imaging data, because so far this dot plot is something what we are getting out of a conventional flow cytometer, and here on the x-axis of this dot plot, on the left side of the plot we are seeing the CD3 positive T cells and the CD14 positive monocytes. So if you look into the image down there, so LFA1 is expressed as green cells, which is expressed by the T cells, and ICAM is generally expressed by the monocytes, right? So now this data from the dot plot, now if you look into the imaging, you will be able to understand that whether an immunological synapse formation is happening or not. Because on the three images, if you look into the first image, you can see there is a yellow light coming in, right, from the middle. So that is the evidence that there is a LFA1 and ICAM interaction has happened, right, and that is giving us the yellow light compared to the passive synapse where we don't see any yellow light coming in. So as this is a direct visualization, direct measurement, it gives you more confidence or opportunity to study the immunological synapses. Same goes for the tumor cell killing assays, which are on the right side. You can see the usually the like if you are working on a TILs or something, some other cell therapy if you are doing, you will be able to see the interaction of the lymphocytes with the cancer cells together. So no indirect measurement, directly you can see the data. Cell-cell interaction studies, as was say translocation studies, what I was telling, any functionality, any kind of pathway study if you want to see, you can do in this way. So nuclear translocation assay, NF-kappa B translocation to nucleus. Conventionally we do it with indirect measurement. Now here on the image wall, as you are seeing, that when in the control or unstimulated, the green one, okay, which is on the left side of the image wall, compared to the right side of the image wall, which is just TNF treated stimulated. So all the right side of the image wall, what you are seeing is a yellow light coming in, and on the left side, which are unstimulated, you can clearly see the nucleus, which is red, and the outside NF-kappa B, which are FITC marked, which is green. So red and green can be clearly distinguished because NF-kappa B is on the cytosol, but with TNF stimulation, what you are seeing in the right of the image wall, all are inside the nucleus, so they are together, so we see the yellow light coming in. So this is a direct measurement for any nuclear translocation or any pathway analysis. If you want to do, you can do in this way. So of course, so far the data I am showing, and this is I'm telling you the advantage of this technology of cell view. Of course, there is an instrument behind it. So this instrument we call FACS Discover S8 Cell Sorter. How it looks like. This is the instrument. It's a benchtop instrument, as you can see. All the laser modules, fluid modules are below the table, and the workstation is there on the top of the table. As I was telling, that what it does, along with the keeping the flow cytometry data integrity as it is adding the imaging information to take the sort decision. In the sorting part, it's a 6-way sort. So it retains all the other benefits of any BD sorter like automated stream setup or automated sorting criteria, but along with it's a 6-way sorter. So you can sort 6 different populations together based on the imaging criteria, based on the flow cytometry criteria, data output, or combined measurement. So this is the correlation, what I say, the benefit further after sorting. So you can take with the cell morphology or the protein expression or CD marker analysis data. If you want to do downstream multiomics analysis with BD Rhapsody system, you can do that after sequencing in, say, single cell mode or maybe in a bulk sequencing. So if you want to sort Treg cell, you can do that based on the imaging criteria, and then you go for multiomics to understand how differentiated further the Treg populations are. So that is how it is connecting to the further other technologies for discovery analysis. And in the CD marker part, the new part which is in, feature which we are giving you, it's a spectral flow cytometer. So it's not conventional. So there are 78 spectral detectors out of 5 lasers which are this equipment is actually having. So 78 spectral detector plus 6 imaging parameters plus FSC and SSC. So that is the total 80 plus detectors what we are getting out of this instrument. And when we are giving you 78 spectral detector, that's a huge number. That's a real high dimensional flow cytometry we are talking about. So we know that we also need to give you the dyes, because the reagent availability is always a question. That we can go high parameter, we can go into 70, 60, 70, 80 plus detectors, but do we have the reagents? So keeping that in mind, BD launched the spectral series of dyes, the BD Horizon Real Yellow, which are laser specific dyes. So if you are conversing with flow cytometry and if you are using fluorochromes like PE, phycoerythrin, you might be aware that it is cross laser excited, like it can be excited by the blue laser as well as the yellow-green laser, which creates sometimes a problem in compensation or data analysis. But all these real dyes, whether it is real yellow or real blue, these are laser specific. So when we call it's a real yellow dye, these are all excited by the yellow-green laser, which is 561, not excited by any other laser. So cross laser excitation problem, which can further contribute to the spread in data, that is eliminated. And along with the image sorting, you can use this spectral information for sorting as well as was mentioned. So this is my last slide, that taking all this together, the cell view image technology, the systematic approach to sorting, spectral analysis and sorting, the software innovation in terms of FlowJo, because when we are bringing in, as I said, this FlowJo lens plugin is also now inbuilt within FlowJo. So if you are using Discover, you are using those, getting those image data, you can use that FlowJo software to analyze. And finally, this combined data is advancing, the sorting module the way so far it is, absent, I would say, in terms of using the real-time imaging for sort decision. So I would like to stop here. If you have any question, I'd be happy to answer.
Thank you, Shobhadeep. He will be available for discussion if anybody has, but if anybody has any very important question, very pressing question, then please ask. I'm sure he has given a lot of information, so many of you may have many questions, but you can discuss during the lunch time. Thank you very much for the information.